previously unknown protein interactions may lead to better HIV treatments. The human immune deficiency virus, HIV, first entered public consciousness in the early 1980s, after cases of unfamiliar and deadly illnesses began to overwhelm medical centers across North America. An estimated 42 million people have died from HIV/AIDS to date, and while people can now live full lives with access to treatments, it is still a chronic condition that people would like a cure for, said Dr. Linda Calico, head of the Department of Microbiology, Immunology, and Biochemistry in the College of Medicine at the University of Saskatchewan, USask. Calico's lab studies immune responses to HIV, particularly a family of proteins called ApoBec3, which cause mutations in the HIV virus. Her research and others show that the virus uses a counterattack that breaks down ApoBec3's defenses, helping HIV win and establish an infection. There is a battle going on in the cell, said Calico. HIV also has a protein known as VIF that it uses against ApoBec3, so it's this fight against each other. Researchers, including Calico, have tried to create drugs that block VIF from interacting with ApoBec3, giving proteins the added boost it needs to fully inactivate the HIV virus and prevent an infection, but these blockers have been mostly ineffective. Now, for decades after HIV was first identified, Calico and her colleagues, including USask postdoctoral fellow Dr. Amit Gaba, Ph.D., have discovered previously unknown interactions between ApoBec3 and VIF proteins, which offer a better map for drug design. Their findings were recently published in the Journal of Virology. Based on our data, previous inhibitors weren't successful because we weren't looking at the right interface between the protein interactions, said Calico. We think that with a better map of this interface, we can design a better inhibitor. Previous studies used one VIF protein and a single ApoBec3 protein to simulate conditions during an infection, but this experimental design isn't quite measuring up to what is actually happening inside our cells. Using a range of isolated HIV viruses, each with slightly different VIF proteins, Calico and her team found that there are differences in the VIF proteins among HIV strains and that ApoBec3s are not working individually against HIV. My lab established that ApoBec3s are working together during an infection, and they form a new structure, said Calico. ApoBec proteins are part of a natural defense system against viruses, and if we can stop that interaction with VIF, they could be a kind of natural cure. According to Calico, this discovery could pave the way for new treatments. The potential for ApoBec proteins to serve as a natural defense against HIV brings a new wave of optimism in the fight against the virus. HIV still remains a global public health issue, said Calico. Current treatments only suppress the virus, so we're really hopeful that our research offers a new avenue, where we can help the body's natural defenses, stopping HIV infections from taking hold.